Dear students, welcome to the Medical Genetic course. Today's lecture is part 1 of non-Mendelian inheritance. Dear student, by the end of this video, you are expected to define genomic imprinting and epigenetics and uniparental disomy. You will also know the definition of dynamic mutation. In today's lecture, you will know other types of inheritance. It's called non-Mendelian because it does not follow the Mendel law of segregation. The first topic is genomic imprinting. Each person has 2 copies of the same chromosome, one from the mother and the other one from the father. For some human genes, one of the alleles is transcriptionally inactive, which means there is no mRNA produced, depending upon the parent from whom the allele was inherited. Although you think that genes on homologous chromosome are expressed equally, but it is actually not, because it's now recognized that different clinical features can result depending on whether a gene is inherited from the mother or inherited from the father. This is called parent of origin. This part of origin effect is referred to as genomic imprinting. It is methylation of DNA is the main mechanism by which expression is modified. If an allele from the father is inactive, and the maternal allele is expressed, then it is called paternal imprinting because the father allele is silent. On the other hand, if an allele from the mother is inactive, which means silent, and the father is expressed, it is called maternal imprinting. The question is, how does genomic imprinting happen? Silencing or inactivation usually happen through a process called DNA methylation. About 80 human imprinted genes were discovered to be imprinted. Imprinting is a normal process caused by alteration in chromatin that occurs in germline of one parent, but not the other. Therefore, one of the allele is transcriptionally inactive, which means that there is no mRNA produced, and is said to be imprinted or silenced. This change is heritable and reversible. Methylation is determined during embryogenesis and passed over to differentiating cells and tissues. A question: what if something goes wrong during genomic imprinting? For example, there is a defective allele from non-imprinting parent, or there is a deletion from non-imprinting parent, or both alleles inherited from parents, which is called uniparental disomy. This will be explained in the next few slides. Let's take an example.